Hello guys, and today our topic is continuation of the renal tubular processing. And today we are going to discuss about the proximal convoluted tubule functioning. Just a little bit of revision from the previous lectures, and we're going to discuss about the loop of Henle, the descending part, and the thin and thick ascending part of the limb of loop of Henle. So let's get started. First of all, we are going to discuss about the PCT. As you know that the proximal convoluted tubule cell has the luminal border that has a brush border. Okay, the brush border is there to increase the surface area for reabsorption approximately 20 times, and on the basolateral surface the proximal convoluted tubule cell has some striations. These striations are nothing but mitochondria because these cells are involving a lot of ATP. They are investing a lot of ATP, so they require mitochondria for that. And these cells are held together by tight junctions. These are tight, but these are not enough tight for the water. These are not watertight. Okay. Along with the tight junctions, the water can pass by the solvent drag. Right? We have discussed that, and there are a lot of sodium potassium ATPases. These are the sodium potassium ATPases on the basolateral surface of the cells, and these constantly pump sodium out and potassium into the cell against their concentration gradient by the primary active transport mechanism. And in this condition, the sodium concentration inside the cell decreases and potassium concentration starts to increase, and there are some leaky channels for the potassium, and the potassium starts to leak from these leaky channels. It makes the cell inside minus 70 millivolt. It makes inside of the cell negative, right? Because sodium has been moving out and potassium also been leaking out, so the inside of the cell becomes even more negative. This negative is a electrochemical gradient for the sodium inside the lumen. Also, the sodium has a concentration gradient and electrical gradient into the cell. It moves into the cell by the facilitated diffusion, but these sodium transporters can bind other substances with them as well, like glucose. They can bind amino acid, they can bind chloride. Okay, they can bind chloride ion. They can transport these in by the secondary active transport, because ATP is not being utilized here directly. Okay, ATP is not utilized here directly. We have discussed all these mechanisms in detail in the previous lectures. And there's also another pump known as the sodium hydrogen or sodium proton anti-transport, because it transports the sodium into the cell and proton out. This proton combines with the filtered bicarbonate and forms carbonic acid, which then dissociate into carbon dioxide and water. This carbon dioxide moves into the cell by diffusion and combines with the water present already inside the cells, again form carbonic acid, and this carbonic acid dissociate to proton and bicarbonate, and this is the bicarbonate that we are making from the filtered bicarbonate by indirect process. This is not a direct reabsorption, it's like indirect processing of bicarbonate. Also, the solutes like sodium also leaked out by the these pumps. Okay, potassium also leaked by the leaky channels. The glucose, amino acids, chloride, all of them are present in the interstitium. So this interstitium become hyperosmolar because there are a lot of active solutes present here, and this kind of hyperosmolarity attracts water from the lumen side, and the water is moved along with the solutes present in the water, like small quantity of sodium, small quantity of bicarbonates or amino acids are moved along with the water. This is known as the solvent drag. Not only these low molecular weight proteins like insulin and hemoglobin in the free form, they're also reabsorbed or captured by the PCT cells by the pinocytosis. It's also a type of active transport in which the clathrin proteins helps to invaginate the membrane. It pinches off in the form of vesicle, and later it then release these proteins, low molecular weight proteins, and then we reabsorb them back into the blood. Also, not only the reabsorption but secretion in the PCT is very important. Substances like para-aminohippuric acid, oxalate, urate, bile salts, organic acids and bases, drugs, toxins are all secreted actively into the lumen from the cells, okay, from the blood. And there is a drug known as probenecid. Probenecid was an old drug that was used before, but the function of probenecid is somewhat different. It is used to increase the action time of penicillin. How does it cause the increase in the action time of penicillin? So, because there are secretory proteins, or we can say the like channel proteins that are secreting the substances from the blood to the lumen of the nephron. And probenecid does what? It blocks that secretory proteins, it inhibits the secretory proteins to function, right? So the secretory proteins are inhibited, so that's why these secretory proteins or secretory channels can no longer secrete the penicillin, and penicillin stays in the blood for a longer period of time. That's why probenecid is used to increase the action time of penicillin by blocking its secretory proteins. Also, hormone influence in the PCT is worth mentioning, that phosphate reabsorption. We haven't discussed phosphate reabsorption. Phosphate reabsorption in the proximal convoluted tubule depends on the hormone PTH. We can remember the parathyroid hormone as phosphate trash hormone. It trashes the phosphate. It does not allow the reabsorption of phosphate. Okay, it promotes the excretion of the phosphate, right? It does not allow the reabsorption of the phosphate. Another function of PCT is that it is involved in the conversion of inactive vitamin D to active form. How does this work? Let us discuss about that. Our skin, when exposed to the sunlight, make cholecalciferol. The cholecalciferol undergoes two processes of hydroxylation, first in the liver and second in the kidneys. This cholecalciferol goes to the liver, and hydrogen is attached to 25th position, right? And 25th 25 hydroxy cholecalciferol then comes to the kidney, and the proximal convoluted tubule cells have an enzyme known as alpha 1 hydroxylase, because there is the process of hydroxylation and it attached another hydrogen to the position number 1, making 25 to 125 dihydroxycholecalciferol. And this 125 dihydroxycholecalciferol is an active form of vitamin D that can go to the GIT and can involve in the calcium reabsorption from the GIT, or calcium absorption. Also, another hormone influence, or we can say substance, is that angiotensin 2. Angiotensin 2 is vasoconstrictor of the efferent arteriole, okay? It's the vasoconstrictor of efferent arteriole. But apart from that, angiotensin 2 can also be involved in the sodium reabsorption. Okay, it's a fact that angiotensin 2, apart from its function of vasoconstriction of efferent arteriole, it can also be used in the sodium reabsorption from the PCT. We are discussing about osmolarity here. What is osmolarity? Osmolarity is like the concentration of the body fluids or the plasma, and it is determined by the osmotically active solutes present in the fluids or plasma. What does it mean? It means that if solutes are more and the solvent concentration is less, so that environment will be called as hyperosmolar. And another environment, if there the solvent is more, means water is more, and solutes like chlorides, sodium, potassium, amino acids are less, so that environment would be called as hypoosmolar environment. Our plasma and body fluid osmolarity is approximately 275 to 295, but we have written an average like 292 milliosmol per liter, and it is determined by the osmotically active solutes present inside the fluids or plasma. What would you think that osmolarity in the proximal convoluted tubule when going towards the descending limb of loop of Henle, does it increase or does it decrease? Let us see about this. Okay, first we are discussing about osmolarity present in the proximal convoluted tubule when filtration occurred from the Bowman space or the glomerulus. The plasma has osmolarity of 300. We can just write 292 as 300 for easy calculation purposes. Okay, approximately 300 milliosmol per liter is present in the capillary. But when the filtration has occurred, what do you think? Osmolarity in the proximal convoluted tubule, does it changes, does it increase or does it decrease? Maybe students can confuse that a lot of solutes are reabsorbed from the PCT. That's why maybe the osmolarity has been increased. But the truth is that absorption of water is also occurring along with the solutes, right? So same amount of water and solutes are being reabsorbed from the PCT. Absorption of water and solutes from the PCT is isoosmotic. It means that same amount of water and same amount of solute has been reabsorbed, so the ratio present in the lumen still PCT is the same as the previous one before the reabsorption. So osmolarity in the PCT is not changed, it's isoosmotic. But there is a fact that nephrons are present in the cortex as well as present in the medulla of the kidney. But as we move from the cortex towards the medulla, the medullary interstitium is more more hypertonic. It means that it is hyperosmotic as compared to the cortex, and we will discuss that later in this lecture. Just now you just trust me that fluid in the medullary interstitium is hyperosmotic. Fluid in the medullary interstitium is hyperosmotic as compared to the cortex. We have discussed proximal convoluted tubule in detail. Let's talk about the descending limb of loop of Henle. As you know that loop of Henle has two parts. It has the um, descending limb, it has ascending limb, and ascending limb has further two parts, the thin ascending limb and the thick ascending limb of loop of Henle. We are talking about the descending limb of loop of Henle, some characteristics of these cells. These cells lack brush border because there is not a lot of reabsorption, because reabsorption has occurred in the PCT. So these cells do not have mitochondria because these are not involved in active transport mechanisms. They lack basolateral striations because they are less metabolically active. And one more very important thing to remember, that this part of the nephron from the proximal convoluted tubule up to the descending limb of loop of Henle, this is under all physiological circumstances, this part of the nephron is freely permeable to water and partially permeable to solutes. What does it mean? It means that the descending limb of loop of Henle under all physiological circumstances is freely permeable to water. But there is another part of the nephron from the ascending limb of loop of Henle up to the distal convoluted tubule, the proximal part of the distal convoluted tubules. These are under all physiological circumstances not freely permeable to water. As we're discussing about this part here, okay, this part dips into the hyperosmotic interstitium, medullary interstitium, okay, because this part is water permeable. So this part I will draw it here for you, that we are talking about the descending limb of loop of Henle, right? And as this part of the nephron dips deep into the medullary interstitium, that is hyperosmotic, and the descending limb of loop of Henle is already water permeable. So here's a lot of solutes present here, right? So what does water inside will follow? The water in the descending limb of loop of Henle will move out towards the hyperosmotic gradient. It has already a gradient established that water need to follow that. It's the property of water that it follows from that it flows from less concentration towards the high concentration of solutes, and some of the solutes from the interstitium will move into the tubule. So what do you think? Here was 300 in the capillary. In the proximal convoluted tubule, we have discussed that osmolarity has not been changed because the reabsorption of water and solutes from the PCT is isoosmotic in all conditions. But in thin descending limb of loop of Henle, as the water has moved out and some of the solutes have gained, so it means that osmolarity in the tubule of the descending limb of loop of Henle is increased, and a point come when the hairpin loop starts to begin forming, right? It's the hairpin loop. The medullary interstitium also has 300. It starts to increase from 300 to 400 and up to 1200 milliosmol per liter. And the point where it starts to twist, it starts to form the hairpin loop. The osmolarity inside the tubule also became 1200 milliosmol per liter. It is balanced by the interstitium. These two fluids, the tubular fluid and the interstitial fluid, starts to equilibrize each other right here in this condition. So it means that its concentration become equal to the interstitial osmolarity, which is equal to 1200 milliosmol per liter. Now we have discussed descending limb of loop of Henle. Let's move towards the ascending limb of loop of Henle. As I have told you, the ascending limb of loop of Henle has two parts, the thin part and the thick part of the ascending limb of loop of Henle. And most important discussion in today's lecture is about this topic. As I have discussed that these both parts are water impermeable, I have drawn this red line to make it more even highlight that the thick ascending limb and the thin ascending limb both are water impermeable under all physiological circumstances. And why is that so? It's due to the presence of tight junctions. Tight junctions were also present in the proximal convoluted tubule, right? We have discussed that tight junctions are also here, but these are not as tight as the present in the ascending limb. These are not watertight. These are not watertight in the PCT, but when we talk about the ascending limb, these junctions are watertight. It's very important. There are a lot of cells present here in the ascending limb, in the thick ascending limb to be more particular. Just we are now going to focus only on one cell, and I will show you how this cell functions, what are the kind of transporters that are present in this cell. So let's discuss about it, okay? First we will see in the diagram and then we will read the text. This is the cell of the thick part of the ascending limb of loop of Henle. As in the PCT cells on the basolateral side, it has a sodium potassium pump or sodium potassium ATPase. It is constantly throwing the sodium out and potassium into the cell via primary active transport mechanism. The concentration of sodium inside the cell starts to increase, okay, starts to decrease, sorry, because this pump is throwing the sodium out and potassium in. The potassium concentration inside the cell is going to increase and the sodium concentration starts to decrease. On the basolateral surface, there is a special kind of transporter present in particularly thick part of ascending limb of loop of Henle, which is known as sodium potassium 2 chloride cotransporter. Sodium potassium 2 chloride cotransporter. The name suggests all of its functions. What are the functions? It transports the sodium in, it transports the potassium in, it transports the chloride in. But what are the mechanisms? Okay, as we know that sodium concentration inside the cell is decreased due to the pumping of sodium constantly from the sodium potassium ATPases. Sodium is moving down the concentration gradient by the facilitated diffusion. Okay, the potassium concentration inside the cell is high, so potassium is being moved by the secondary active transport, and the chloride also moved by the secondary active transport mechanisms. But the sodium here inside the cell move out by the primary active transport. The potassium built inside the cell has some leaky channels that can be moved out, and chloride also has some its special channels and they can be moved out into the cell, making the interstitium hyperosmolar. And this is the point what I wanted to mention, why I said that medullary interstitium is hyperosmotic. We have discussed, right? The osmolarity from the cortex to medulla starts to increase. It is because that these pumps present on the both side of the tubule, they are functioning in transport of solutes from the lumen through the like transcellular route into the interstitium. They are constantly throwing the solutes into the interstitium, making the medullary interstitium hyperosmotic, and that's the reason why osmolarity increases from cortex to medulla. And now you know the reason as well. But there is something very important in case of potassium, that potassium is increasing in a very dangerous amount inside the cell, because this transporter, the sodium potassium ATPase, is also transporting potassium in, and this transporter, sodium potassium 2 chloride cotransporter, is also transporting potassium into the cell. So potassium is dangerously increasing inside the cell, so it needs the way to escape out of the cell. So what are the two ways that the potassium can escape through? First is the basolateral side from the leaky channels, right? And these leaky channels are not only present on the basolateral side of the cell, but these are also present on the luminal side of the cell. And when the potassium move from the cell into the lumen again, this process is occurring again because potassium has once been transported into the cell and now it starts to move into the lumen. As potassium is a cation, it makes the luminal membrane, or the membrane that is close to the cells, or we can say the membrane, more electropositive because it's a cation, right? So potassium is being pumped out of the cells on the both side of the thick part of the ascending limb of loop of Henle, and it makes the cell, it makes the membrane electropositive, approximately 8 millivolt. As you know that solutes and the tubular fluid is moving like here. Okay, we have only discussed a small portion in this diagram. This is the thick ascending part of the loop of Henle, and this is the cell of that. But here the whole tube continues. The tubular fluid is coming, and this tubular fluid contains calcium and magnesium. Calcium and magnesium are also cations. As this membrane become electropositive, these cations are kicked out from the lumen into the interstitium via the paracellular routes, like they can escape between the cells because they do not find the way through the cell, they just escape in between the cells. This is the way by which calcium and magnesium are reabsorbed in the thick part of the ascending limb of loop of Henle. So let's summarize what we have discussed, that the cells of the thick part has sodium potassium ATPase on the basolateral surface. They pump the sodium out, they form a gradient. On the luminal surface, they have sodium potassium 2 chloride cotransporter. Okay, and 25% of sodium is reabsorbed down its concentration gradient. Why have written down the concentration gradient? Because sodium inside the cell is already low. Lumen, it's high, so it's moving down the concentration gradient. And absorbed chloride will move into the interstitium by leaky channels. Absorbed potassium will leak into the interstitium. Okay, but potassium will also leak by the leaky channels again to the lumen, and this leaking of the potassium into the lumen make the lumen electropositive. about 8 millivolt, and this electropositive charge repels the cations in the tubular fluids like calcium and magnesium out of the lumen into the interstitium via paracellular routes. This is the mechanism by which the calcium and magnesium are reabsorbed. And a lot of solutes are present in the interstitium, right? The chloride, the potassium, calcium, magnesium, sodium. So this automatically makes the medullary interstitium hyperosmotic. That's why this part, the thick ascending limb of loop of Henle, is making the medullary interstitium hyperosmotic and the tubular fluid hypoosmotic, because all of the solutes have been moved out and the osmolarity starts to decrease from 1200 to only 100. Okay, that's like a logical explanation to what has happened right now. Last topic for today's discussion is loop diuretics. Loop diuretics are the drugs, for example, furosemide, bumetanide, torsemide, and atacrinic acid. These are used in the diuresis purposes. Okay, these block the function of sodium potassium 2 chloride cotransporter. When these loop diuretics block the function of sodium potassium 2 chloride cotransporter, sodium cannot be reabsorbed, the potassium cannot be reabsorbed, and the chlorine cannot be reabsorbed. And apart from reabsorption, they are excreted along the drain. And the second important part in the function of loop diuretics is that when the potassium is not pulled into the cell, the potassium concentration inside the cell does not increase, and this potassium does not leak out into the lumen again. It does not make the membrane electropositive. When the membrane of the ascending limb of loop of Henle does not become electropositive, so how it's going to kick the back of cations like calcium and magnesium? And the calcium magnesium reabsorption also stops by taking loop diuretics. And does it have any clinical implications? Yes, it have. The loop diuretics are contraindicated in case of women with osteoporosis, because those women already have less calcium inside the blood, and loop diuretics promote calciuria by inhibiting the reabsorption of calcium and magnesium. So in case of osteoporosis, loop diuretics are contraindicated. So this was for today. I know this was a little long lecture, but I hope it worth it for your knowledge. I will see you in the next video with other discussion about the renal tubular processing, the distal convoluted tubule, and